helps to treat anemia. Okay, next is asthma. Interleukin 1 receptor. This is used to treat asthma. Next is atherosclerosis. What is atherosclerosis? It is narrowing of coronary arteries. Okay, so platelet derived growth factor is released, and this platelet derived growth factor dissolves these plaques if present inside coronary arteries and thereby treats atherosclerosis. Parturition, relaxin, which is naturally released from corpus luteum. Okay, so in absence of relaxin, parturition, externally relaxin is essential. Okay, and that is injected. This relaxin is obtained biotechnologically. Blood clots, they can be treated by tissue plasminogen activator TPA and urokinase. So these TPA injections are given if some blood clots are moving through arteries and capillaries, arteries especially, they may block somewhere in capillaries. Okay, so to dissolve these clots, this TPA, that is tissue plasminogen activator, is injected. Cancer interferons. What are interferons?
Basically, the author cells that the virus is
But
infecting.
the cells are dying. The cells which release interferon, they are already these dying. So these cells are affected by viruses. These cells are dying, but they release interferons to give signals to adjacent cells and make them protective against the viruses. Okay, so naturally they are released. Artificially they can be produced biotechnologically. Tumor necrosis factor, this can be used to reduce or to undergo atrophy of tumor. Interleukins, macrophage activating factor. Next, diabetes, insulin, emphysema, alpha antitrypsin. What is emphysema? कशाच्या? रेस्पिरेटरी सिस्टमचे डिसऑर्डर आहे. एम्फायसेमा म्हणजे गुदमरल्यासारखं होतं. विंड पाईप जरा चोक होतो आणि त्यामुळे व्हेंटिलेशन प्रॉपरली होत नाही. दॅट इज एम्फायसेमा. सो अँटीट्रिप्सिन इज गिवन. नेक्स्ट, hemophilia A, blood activating clotting factor 8, hemophilia B, blood clotting factor 9, hepatitis B vaccine. This list you have to remember. Understood? Okay. So healthcare biotechnology, it refers to a medicinal or diagnostic product or a vaccine. This technology has tremendous impact on meeting needs of patients. Biotechnology offers patients a variety of new solutions such as unique, targeted, and personalized therapeutic and diagnostic solution for organ transplant. We know that in organ transplant, major problem is organ of donor and recipient, their antigens do not match. Okay, so in this way we can give specific antigen coated organs which can be accepted easily in the recipient's body. Okay, so they are called as personalized solutions. Stem cell technology, so stem cells are extracted, they are cultured, they are triggered to produce some type of tissues like skin or maybe liver cells or maybe neurons or maybe cardiac muscles. Genetic counseling, so some genetic disorders can be treated. Some faulty genes can be replaced by healthy genes, or unwanted carcinogenic genes can be removed. Okay, forensic medicine, gene probes, genetic fingerprinting, and karyotyping are outcomes of biotechnology. Okay, what are gene probe? Gene probe are small segments of DNA which are used to identify their complementary DNAs present in entire genome. Okay, so these small sequences which are synthesized, which will identify their complementary DNA sequence, these are called as gene probes. Okay, हे सगळं समजलं का? बघा एकदा वाचून हा पॅराग्राफ. Go through this paragraph. In your theory exam, they will not ask how genomics is used as solution of our organ transplant, how it is used in stem cell technology. Such questions they will not ask. But you must know meaning of these terminologies. Done? Chalo, next. Human insulin. Insulin is a peptide hormone produced by beta cells of islets of Langerhans of pancreas. It was discovered by Sir Edward Sharpe Schaeffer in 1916 while studying islets of Langerhans. Insulin is essential to control blood sugar level. Diabetes mellitus is a disease in which some people cannot make insulin themselves. Hakura et al. In 1977 chemically synthesized DNA sequence of insulin for two chains A and B separately inserted into two plasmid PBR322 vector. Okay, so what they did, see insulin, this hormone has two peptide chains. One is A and other is B. ओके एंड ए का जीन एक व्हाट्स क्रोमोसोम पर है, बी पेप्टाइड का जीन जो है वो अदर क्रोमोसोम पे है. ओके सो दिस हकुरा एट al what they did, they took out a gene for a polypeptide and exactly same gene they inserted in PBR322 and B peptide ka gene they inserted in another plasmid PBR322. Okay. Insulin production by recombinant DNA technology is designed by Gilbert and Willow Markov in 1978. The genes are inserted by the side of beta galactosidase gene of a plasmid. So if beta galactosidase त्याच्या शेजारी जर जीन इन्सर्ट केला, that means beta galactosidase will not be functional, and we studied that blue color of cells will go, right? The recombinant plasmid were then separately transformed into E. coli host. The E. coli host produced penicillinase positive pre-proinsulin. Insulin is later separated by trypsin treatment. Okay, so penicillinase plus pre-proinsulin. Penicillinase, this is enzyme which is required. This enzyme will act as marker gene, and pre-proinsulin, that is precursor of insulin, is produced, and this insulin is then separated by trypsin treatment from E. coli cells. So E. coli cells are producing insulin. Fine. Next, vaccine production. A vaccine is a biological preparation that provides active acquired immunity against certain disease. Active means our body synthesizes antibody. Acquired, it is acquired in life, immunity against certain diseases. Usually a vaccine consists of a biological agent that represents a disease causing microorganism. It is often made from a weakened or killed form of microorganism that is called as attenuated. It's a toxin or one of its surface protein antigen. We have studied all this, right? Okay, sometimes toxins which are harmful for us, these toxins are extracted, they are called as toxoids, and then these toxoids are mixed with some other chemicals and injected. So TT का vaccine जो होता है actually it is a toxoid. Okay, tetanus का. ठीक है. All other microorganisms are hit killed or weakened, that is called as attenuated microorganisms, and these attenuated weakened microorganisms are injected in body, against which our body prepares antibodies. Okay, so naturally this is how vaccines are prepared. Biotechnology has offered modern diagnostic kits for rickettsia, bacterial and viral vaccines along with radio-labeled biological therapeutics for imaging and analysis. Okay, so these vaccines are radio-labeled, so they can be identified from the organism in which they are injected. So imaging and analysis, how this vaccine is affecting, that can be analyzed easily. Vaccines have eliminated smallpox, polio, and other deadly diseases for last several decades. Biotechnology has made advancement in vaccinations by making recombinant vaccines that have potential to eradicate non-communicable diseases like cancer. Naked DNA vaccines. Naked DNA vaccines means only DNA without any protein coat or membrane covering. Viral vectors vaccine. Okay, so viruses which are carrying DNA, and this DNA will provide us immunity for a particular disease. So they are viral vectors vaccines, and plant derived vaccines are found to be most effective against a number of bacterial and viral disorders. So naked DNA vaccines means only DNA fragment is given injected in our body. Viral vector vaccines means these viruses which carry DNA segment, but these viruses are made harmless, and these viruses are allowed to infect host organism, so that the DNA segment which will help us to get immunity can be injected. प्लांट डिराइव्ड वैक्सीन, इसके लिए हमने पोटैटो का एग्जांपल लिया था. इट इज गिवन इन नेक्स्ट पैराग्राफ. आई विल रीड दैट. Okay, oral vaccines, a novel approach. You know that all vaccines are injected. Okay, there are few vaccines like swine flu ka vaccine jo hai, they put drops of that vaccine in nose. Okay, so vaccine ka basic principle kya hai, that vaccine should enter in bloodstream. Okay, so bloodstream में immediately जाना चाहिए, so that it will remain in same chemical form and it will trigger our immunity. Okay, why vaccines or insulin is not given orally? Because in stomach or intestine it will get digested and chemically it will get degraded, so it will not remain vaccine anymore. Okay, but in oral vaccine, अब इंजेक्शन का जरूरत नहीं है. Orally some vaccines can be given and they will remain in the same chemical form. Okay, what is oral vaccine? The latest hotspot in the field of vaccine research is the development of oral vaccine which can be taken orally. Immunogenic protein of certain pathogen is found to be active when administered orally. The gene corresponding to such proteins is isolated and gene construct is produced. Okay, so the gene which produces this protein, that gene is isolated and that gene is inserted. This is introduced and expressed in plant genome. Okay, so वो gene निकाल के plants में डाल दिया है. Okay, and in plant cell it will get incorporated with the genome of plant. Okay, which results in production of such immunogenic protein in parts of plant where it is expressed. So suppose in leaves it is inserted, then leaves will produce that protein. Okay, these when fed into animals or mainly humans, the person becomes vaccinated against certain pathogen. The only problem is it should be consumed raw. Okay, so in potato they have tried to insert some vaccine. Okay, but potato should be eaten raw so that the protein will remain protein, it will not get degraded. Okay, but raw potato we will not consume. Okay, we will always at least boil or roast them. So उसके बाद वो protein जो है वो protein degrade हो जाता है. Such vaccines are known as edible vaccines. Okay, otherwise vaccinated potato का we would have made batata vada na. Okay, but that protein gets degraded. ठीक है, सो इट हैज सम लिमिटेशंस. अभी मार्केट में सारे वैक्सीन ये प्लांट के फॉर्म में नहीं है. ठीक है, एन एक्साइटिंग इन्वेंशन is production of melt in mouth vaccine that can be administered by placing them under your tongue that delivers it into bloodstream. Below tongue, whatever mucosa is there, it has capillaries which are superficial. Okay, you can even observe below tongue, you can see small network of capillaries. Okay, so some gel or small pieces of gel are prepared in which there is a vaccine, and these pieces of gel can be placed below tongue for some time so that it will get dissolved and that vaccine will get absorbed in the blood capillaries present below the tongue. Okay, so that is one kind of oral vaccine where vaccine did not reach our stomach and intestine, but it will get absorbed in the capillaries present in the thin capillaries of mucosa. Okay, the most important example is production of flu vaccine by bacillus, which melts in mouth. The tremendous benefit of such vaccines is the comfort of administration. लो कॉस्ट अँड इज ऑफ स्टोरेज. कम्फर्ट ऑफ ॲडमिनिस्ट्रेशन हा सगळ्यात मोठा पार्ट आहे. ओके, नाहीतर रडणाऱ्या ओरडणाऱ्या मुलांना पकडण्यातच सगळा जीव जातो डॉक्टरांचा. फाईन. नेक्स्ट, ॲग्रिकल्चर. Application of biotechnology in agriculture involves specific techniques such as genetically modified organisms. BT cotton, this we have studied in enhancement in food production, right? What is BT? BT stands for what? Bacillus thuringiensis, right? Just a minute, huh? So BT is Bacillus thuringiensis, pest resistant plants. It helps in modifying plants, animals and microorganisms and improve their agricultural productivity. Who left? Everyone is there, no? Okay. So BT cotton, pest resistant plants. So in plants, naturally, if these characteristics are missing, then gene which is responsible for pest resistance, or let's say genetically modified organism, means for production of certain vitamins or proteins, then these genes are extracted from some organism and inserted in plant, so that plant will have this add-on gene with them, which will help them to produce better variety of crops. We have studied golden rice ka example like this in enhancement in food production. That golden rice, which is rich source of vitamin A in genetically modified plant. Okay. So it can be introduced in microorganisms also to improve their agricultural productivity. How microorganisms will help to improve agricultural productivity? Bola. How microorganism will help to produce agricultural productivity? मधुरा, आर्या, यश, रुद्र.
irrigation system चांगला करायला जाते.
हा.
Irrigation systems जास्ती improve करायचे.
इरिगेशन.
हो, agricultural product.
Microorganisms म्हणतात आपण. How microorganisms will help to produce agricultural productivity?
ते nitrogen fixing bacteria.
Correct, correct. Okay. So some bacteria which are not nitrogen fixing, we can make them nitrogen fixers so that they will improve agricultural productivity. Understood? Very good, Arya. Tissue culture is used in micro propagation, that is large scale propagation of plants in very short duration. Tissue culture technique is also best method of storing germplasm and maintaining a specific genetic type, that is called as clones. So we have discussed this micro propagation. Take out apical bud, that is called as explant, sterilize it and put it in agar gel or artificial nutrient medium, okay, in sterilized condition, and allowed it to grow. So it will form callus. This callus can be cut in many pieces. Each piece can be transplanted to new culture medium, and each, if given treatment of auxin and gibberellins, it will produce roots and shoots and small plant leaves. Okay, so all these plants produced will be clones of each other. So genetical clones of plants can be produced, and this is nothing but tissue culture or biotechnology. Okay. This technique is used in those plants which produce recalcitrant seeds or produce highly variable seeds. Okay, recalcitrant seeds means they require specific conditions. Recalcitrant seeds means reduction in seed moisture content below certain level, and freezing drastically reduces survival and thus present difficulty in storage. Here, subcellular damage of seeds occur, accompanied by consequent loss of viability when dried. Okay, so these seeds are useless to sow and produce new plants. Okay, so seeds तो बनता ही नहीं है. Then how these plants naturally produce? They undergo asexual reproduction. But when you want to extract them from their natural habitat, you want to grow them in new habitat, then tissue culture will help initially to increase their clone number, and later on you can slowly transport them to local conditions. Okay. And highly variable seeds means you are not getting consistently same genetic makeup. Okay. Every time plant has some different characters. If you want some specific characters in next generation, then seeds will not maintain those specific characters, then you need to produce their clones by biotechnology or tissue culture. Okay. Next, gene therapy. A gene is a stretch of DNA required to make a functional product such as a part or all of a protein. Okay, part of protein means what? You know that hemoglobin is made up of heme plus globin, and globin protein is made up of two alpha chains and two beta chains, right? The gene for alpha chain is located on 16th chromosome, and gene for beta chain is located on 11th chromosome. Okay, so the gene which is present on 11th chromosome produces a part of hemoglobin, not entire hemoglobin. A gene which is present on 16th chromosome produces a part of hemoglobin and not entire hemoglobin. Okay, so some genes produce parts of some proteins, whereas some genes, their product, that is polypeptide, produces entire protein. Is it clear? Okay. During gene therapy, DNA that codes for specific genes are delivered to individual cell in the body. Okay. So हर एक सेल को हमें ट्रीट करना होगा, right? Gene therapy is treatment of disease by replacing, altering, or supplementing a gene that is absent or abnormal and whose absence or abnormality is responsible for a disease. So, किसी भी disease, if the disease is because of one gene, that one gene can be replaced with healthy gene, that one gene can be removed. Okay, so वो जो gene में alteration करना है, that can be done to treat the disease. But remember, each and every cell will need the replacement of gene. Okay. Most, if not all, diseases have a genetic factor. Okay. So many diseases which are inborn diseases have some, many times they have some genetic basis. The genetic factor can be wholly or partially responsible for the disease. For example, in disorders such as cystic fibrosis, hemophilia, and muscular dystrophy, changes in a gene directly results in the condition. Highlight this. This you have to remember. एक ही जीन में कुछ ऐसा अल्ट्रेशन है which is responsible for these diseases. Cystic fibrosis, hemophilia, and muscular dystrophy. You know all these diseases. In cystic fibrosis, some fibroids are produced. Hemophilia, blood clotting factor नहीं है, so blood का clotting slow होता है या होता नहीं है. Muscular dystrophy, muscle undergoes atrophy and therefore there is weakening of muscle. In other conditions such as high cholesterol and high blood pressure, genetic and environmental factors interact to cause the disease. Okay, so sometimes there is genetic root, genetic reason, but environmental factors means what are the conditions, what is the lifestyle of the person, that affects more. Okay, so both are responsible for those diseases. There are more than 5,000 different human genetic diseases known to be caused by single gene defects like sickle cell anemia, thalassemia, Tay-Sachs disease, cystic fibrosis, Huntington's chorea, hemophilia, alkaptonuria, albinism, etc. Okay, so sickle cell anemia, you know, thalassemia, alpha and beta thalassemia. This is quantitative disorder of globin. Tay-Sachs disease, Tay-Sachs disease में क्या है? There is one क्रीडू चैट इज फिफ्थ क्रोमोसोमल आर्म इज ओपन. Tay-Sachs disease क्या है? देखो, cystic fibrosis we studied, Huntington's chorea, hemophilia, you know, blood clotting, alkaptonuria, these urine में these abnormal chemical is released. Albinism, you know, no melanin synthesis in any cell of the body. Tay-Sachs मिला. Tay-Sachs disease is a rare genetic disorder passed on from parent to child. It is caused by absence of enzyme that helps to break down fatty substances. These fatty substances, called as gangliosides, build up to toxic level in brain and spinal cord and affect function of nerve cells. हो घेतली. Okay. Huntington's disease is rare inherited disease causes progressive breakdown of nerve cells in brain. Huntington disease has a wide effect on person's functional abilities, usually results in movement, cognitive and psychiatric disorders. No one will ask you what are symptoms of these diseases, huh? Alkatonuria, where body cannot produce what? Urine turns dark brown or black when exposed to air. Okay. So in diseases के symptoms टेक्स्ट बुक में नहीं दिया है. No one will ask you symptoms of these diseases. Okay. Gene therapy is used in many ways. For example, replace missing or defective gene. So replace it with healthy gene. Next, deliver genes that speed the destruction of cancer cells. Okay, so some cancers are injected with some genes. These genes are responsible for atrophying that tumor, and they will kill these cancerous cells quickly. Supply genes that cause cancer cells to revert back to normal cells. Okay, so somehow this cancerous cell, whatever mutation is there, should revert and it should turn normal. Deliver bacterial or viral genes as a form of vaccination. Deliver DNA to antigen expression and generation of immune response. So this DNA will produce antigen. These antigens will trigger our immunity to produce antibodies. Okay. Supply of gene for impairing viral replication. So some genes which will act on viruses, and virus which is multiplying in the body, for example AIDS virus, this virus replication will be altered, it will be slowed down and viral diseases can be treated. Provide genes that promote or impede the growth of new tissues. Okay, so in case of burns, some genes will trigger epithelial cells to undergo mitosis at a faster rate. Okay, deliver genes that stimulate the healing of damaged tissue, etc. Okay. Next, I'm reading blue box from next page. Delivery of genes into cells. Genes can be delivered by three ways. Ex vivo delivery, where cells are removed from the patients and the gene is introduced using a viral or non-viral vector, such as Parkinson's disease, a neurological disorder. Okay, but I don't know what is the effect when the cells are already taken out of the patient and they are given the treatment. Then do they inject tissue back in the patient? That I don't know, in this ex vivo. In vivo means जहाँ पे है वहीं पे टिश्यू में we will inject the injection. In vivo delivery, where therapeutic genes are directly delivered into the cells at the target sites of diseased tissue in patients, like intravenous infusion genes to treat cancer are injected directly into the tumor. Okay. Next, use of virosomes, that is liposome plus inactivated HIV virus. So inactivated virus will act as a vector. Okay. Bionic chips are the other methods of gene delivery. So these bionic chips are put in on the tissue or body, which will carry the DNA gene inside body. So some forms of gene therapy, last time we said that if some genes are defective in a person, okay, and if we replace all those genes in the body of that person, then the effect or whatever the advantage is to that organism. But gametes have same defective genes, So with reproduction, same defective genes will get inherited in successive generation. Okay, so either we can treat somatic cell or gamete forming cells, that is germ cells. If we treat germ cells, then the inheritance in successive generations can be controlled. If we treat somatic cell, then that particular organism will get the treatment. Okay, so likewise there are two types of gene therapy. One is somatic gene therapy and other is germline gene therapy. Okay, so germline gene therapy, in this method healthy genes can be introduced into germ cells like sperms, eggs, early embryos. Early means few celled up to morula stage. It allows transmission of modified genetic information into the next generation. Though it is highly effective in counteracting the genetic disorder, it is not encouraged for applications of human beings due to variety of technical and ethical reasons. Okay, so how to replace single gene from a sperm or maybe a ovum? Okay, after replacing the gene, that ovum or sperm should remain viable. Then it will we will have to carry out its in vitro fertilization. After that it should survive and carry all healthy genes normally. Okay, उसका normal सारा growth होना चाहिए. So technically this is not possible and there are some ethical issues also. Okay, so germline gene therapy is limited in humans. Somatic gene cell therapy, in this type of gene in this type of therapy, gene is introduced only in somatic cells like bone marrow cells, hepatic cells, fibroblast endothelium, and pulmonary epithelial cells, central nervous system, endocrine cells, and smooth muscle cells of blood vessels, walls, etc. Modification of somatic cells only affects a person being treated, and the modified chromosomes cannot be passed on the future generation. Somatic cell gene therapy is the only feasible option, and clinical trials have already employed for treatment of acquired disorders such as cancer, rheumatoid arthritis, blood disorders including SCID, Gaucher's disease, familial hypercholesterolemia, hemophilia, phenylketonuria, cystic fibrosis, sickle cell anemia, Duchenne muscular dystrophy, emphysema, and thalassemia. तुमच्या बुकमध्ये पण हेच लिहिलंय. Modified chromosomes cannot be passed in future generation. Okay, so they are talking about future generations of that cell. जिस सेल में हमने जीन ट्रांसप्लांट किया है ना, एक्सचेंज किया है ना, if the cell undergoes division, then successive cells will carry modified gene now. So successive generation मतलब next family के generation नहीं, generation of that particular cell. Understood this? In exam they may ask you to write a note on gene therapy, either germline therapy or somatic gene therapy. How biotechnology is useful in advancement of agriculture. How biotechnology has helped healthcare. आणि हा जो टेबल आपण वाचला ना 12.7, this can be asked for 1 mark or match the following or complete the analogy like questions. ठीक है. Next part, genetically modified organisms, GMOs. These are living organisms whose genetic material has been artificially manipulated in laboratory through genetic engineering. Now, why we need to change genetic makeup of these organisms? Why we require these genetically modified organisms, that is given in next part. This creates combinations of plants, animals, bacteria and viruses genes that do not occur in nature or through traditional crossbreeding methods. Okay, so जो example हमने देखा golden rice का, so naturally vitamin A gene is absent in rice. Artificially we have put that gene in rice plant, and now rice plant is producing vitamin A along with regular carbohydrates. Okay, so naturally the genes which are not there in organisms, we are making these organisms recombinant with the gene we are interested in. Okay, Most GMOs have been engineered to withstand direct applications of herbicide and or to produce an insecticide. However, new technologies are now being used to artificially develop other traits in plants such as resistance to browning of apples. Okay, so apples ka shelf life will be more, and to create new organisms using synthetic biology. Despite biotech industry promises, there is no evidence that any of the GMOs are currently available in market, offer increased yield, drought tolerance, enhanced nutrition, or any other consumer benefit. Okay, so market में जाके if you try and search golden rice, it is not available because the cost of golden rice is so high that instead of that we can simply buy papaya as a vitamin A का source. Okay, so transgenic plants. The human race is very dependent on agriculture, and as world population continues to expand, there must be continuous reassessment of agricultural practices to optimize their efficiency. By the way, few days back, that what is that? Um, Indian Green Revolution ka person expired, na? Swaminathan, Dr. Swaminathan. You read that? डॉक्टर स्वामीनाथन नाव तर ऐकलं ना तुम्ही स्कूलपासून तुम्ही करताय. ही इज पायनियर ऑफ ग्रीन.
Information
हा
material.
ग्रीन रिव्होल्यूशन, पायनियर ऑफ ग्रीन रिव्होल्यूशन इन इंडिया. That person died few days back. Since early times, human beings have sought to improve the quality and productivity of agriculturally important plants. This was done by selection and traditional breeding procedures that were painstakingly slow and difficult. Traditional breeding programs involve sexual crosses of these plants, which resulted in high quality of present day food plants such as wheat, rice, potato, etc. So, Mendelian practices were carried out initially to improve agricultural plants. Okay, so उसमें you have to select male and female plant, artificially cross them, screen the F1 hybrids. Okay, allow them to undergo self pollination or cross pollination. Then okay, but it is time consuming, and you are playing only with the genes which are naturally present in those plants. Okay, genes which are not there naturally, and if you want to introduce them, then we should rely on biotechnology only. Okay, more recently, biotechnological approaches have been applied to these plants to create genetic variations that are beneficial to mankind. First transgenic plant produced was tobacco. More than 60 transgenic dicot plants and several monocot plants like maize, oat, rice, wheat are known. Tomato, soybean, potato, sugar beet, grapes, brinjal, cotton are other transgenic plants. Transgenic plants are being looked up as bioreactors for molecular farming, that is for production of novel drugs like interferons, edible vaccines, antibodies, amino acids, immunotherapeutic drugs, etc. Okay, so these transgenic plants come purpose क्या है? Some medicine, some vaccines should be given through these plants. Okay, what are advantages of genetically modified plants? The way in which one thinks that genetically modified plants can help as first insect pest resistance. It can help farmers to reduce their use of chemical pesticides, which in turn can reduce the cost of producing food. However, an alternative has been available for more than 30 years, which is a biological insecticide from bacterium Bacillus thuringiensis. However, the use of Bacillus thuringiensis spray is limited because of its low stability of the protein in the field. So after spraying this Bacillus thuringiensis, its effect remains for some time, and again after few days you will have to spray this Bacillus thuringiensis. So now what they have done? They have extracted the gene which is responsible for this insecticidal property. That gene they have extracted from Bacillus. They have put it in Thai plasmid, that is tumor inducing plasmid, and this type plasmid is allowed to infect the plant. ओके, सो ये जो प्लांट है, ये प्लाज्मिड जो है वो इंसेक्टिसाइडल प्रोटीन का जीन लेके प्लांट में जाता है. दैट प्लांट लीफ प्रोड्यूसेस इंसेक्टिसाइडल प्रोटीन, एंड व्हेन कैटरपिलर eat few leaves of this plant, the insecticidal protein is released in the gut of that caterpillar, and caterpillars get killed. Okay, so further loss is prevented, and every time we need not spray the bacillus thuringiensis in crop field. Okay, so we have genetically introduced the gene of bacillus thuringiensis in the plant. You understood this or not? Insect resistant plant contains either a gene from Bacillus thuringiensis or a cowpea trypsin inhibitor gene. The genes called cry gene are present in Bacillus thuringiensis, produces a protein that forms a crystalline inclusions in the bacterial spores. When ingested by susceptible insect, a combination of high pH in gut and enzyme proteinase of insects midgut processes them hydrolytically to release core toxic fragments. The effect of these fragments is seen within minutes of ingestion, beginning with midgut paralysis and ending with disruption of midgut cells of the insect larvae. Bt toxin activity has been against many species of insects with the order Lepidoptera, Diptera, and Coleoptera. So these are orders of insects. Order म्हणजे classification मधले order. So all insects which belong to these orders are mostly pests for crop plants, and these Bt toxins are effective against all these organisms which belong to these orders. Okay. So golden rice, a transgenic food crop used to reduce vitamin A deficiency disease. Similarly, a gene alpha amylase inhibitor has been isolated from adzuki bean, that is Phaseolus vulgaris, and transferred in tobacco, and this gene works against pests like Zabrotis subfasciatus and Calosobrucus chinensis. Okay, why they want to protect tobacco plant against the insect pest ones? Okay, so remember this cry gene, plasmid. Okay, next improve nutritional quality. So first हमने देखा genetically modified plants which are resistant to insect pest. Second modification is their nutritional value is increased. Okay, so transgenic plants have also been produced to provide functional food and nutraceuticals for millions of people in developing countries. Rice is the main item in their diet. Because rice does not contain many essential nutrients, malnutrition is common in those countries. Especially terrible is the blindness that results from lack of vitamin A. This vitamin is abundant in milk and in vegetables such as carrots, which most of the poor people in the world cannot afford. I don't know why carrot is not affordable. Golden rice is affordable and carrot is not affordable. Okay. To solve this problem, Swiss researchers created transgenic rice, that is golden rice, and transgenic mustard, that is golden mustard, varieties with high in vitamin A. The golden color is due to vitamin A. They hope that this rice, if grown and eaten in developing countries, would reduce the diseases associated with vitamin A deficiency, that is VAD. Okay, so some transgenic crops produced by functional food and nutraceuticals are substance provitamin A. Potential benefit, it acts as antioxidant present in rice crop, and the gene transgene which is used is phytoin synthase and lycopene cyclase. Vitamin E substance which acts as antioxidant in canola plants. It contains delta, no, this is what gamma tocopherol methyl transferase. Flavonoids, which are antioxidants, are present in crop tomato, and the gene is chalone isomerase. Fructans, which are low calories, present in sugar beets, and the gene is 1 sucrase fructosyl transferase. Iron, its benefit: ion fortification present in rice, and the gene is ferritin, metallothionein and phytase. अब तक तो ये चार्ट पे कोई क्वेश्चन एग्जाम में आया नहीं है, बट आई कैनॉट प्रॉमिस कि इस पे क्वेश्चन नहीं आएगा. Improvement in oil content and oil quality of oil crops such as soybean, oil palm, rapeseed and sunflower have been achieved by transfer of Arabidopsis gene. Okay, so Arabidopsis thaliana is small worm, or the gene is extracted from this Arabidopsis and put in these seeds. Iron deficiency is also serious nutritional problem affecting an estimated 30% of world population. For production of transgenic crops that will produce iron, food rich in iron and iron storage protein ferritin is targeted. Ferritin is found in many animals, plants and bacteria. The gene for ferritin protein isolated from soybean and fasciolas have been transferred to rice. So fasciolas म्हणजे शेंगा असतात ना, घेवडा वगैरे तशा तसे ते फॅसिओला असतात. So from fasciolas and soybean the gene is extracted and transferred in rice. Okay, I am reading the blue box. Genetically engineered herbicide tolerant plants are developed, as in maize, wheat and many other monocot plants. Genetically engineered disease resistant plants against bacterial and viral pathogens are also developed in plants like tomato, potato and tobacco. Plants deficient in amino acids like methionine, lysine and tryptophan are genetically engineered by introducing genes from other sources so as to make seed proteins rich in those amino acids. Leguminous plants like pulses and maize. Okay, so methionine, lysine, tryptophan agar kam hai, now they are genetically modified so that they will be rich for these amino acids. Similarly, genetically engineered plants tolerant to abiotic stresses such as high temperature, water, cold, etc. Water is water is stress, cold, etc. are also developed. We are still in plant biotechnology. So first we said that plant biotechnology to increase pest resistance. Second, plant biotechnology to increase nutritional quality of plant. Now third, modification of post-harvest characteristics. So whatever plant products are, they should remain viable for longer period. That is called as post-harvest modifications. Okay. Diseases and pests bruising the soft fruits and vegetables, heat and cold storage, over ripenness, loss of flavors and odors, etc. lead to great deal of losses during storage and transport of crops. You must have observed that tomatoes have shelf life outside a refrigerator, of course, of only 4 to 5 days. Okay, bananas, they have shelf life lesser than that. Okay, so because of less or short shelf life, wastage बहुत होता है. Okay, so now genetically modified plants will have better shelf life so that wastage will be reduced. Okay, most of these physiological changes are due to endogenous enzyme activity. Okay, so over ripening से भी fruit का taste change होता है. Have you observed in mangoes that over ripened mangoes are not good in taste? Okay, so ये over ripening क्यों होता है? Because some enzymes are active. Okay, so ये enzyme का activity अगर हम slow down करेंगे तो over ripening can be avoided. Okay, genetic engineering has made it possible to slow down these activities. In the tomato, the enzyme underlying this polygalacturonase breaks down the cell wall constituent pectin. You know that cell wall या फिर intercellular lamella जो होता है, it has calcium pectate. Okay, and the enzyme polygalacturonase digests this pectin and therefore सेल का जो कनेक्शंस होता है वो कनेक्शन थोड़े वीक हो जाते हैं एंड देयरफॉर फ्रूट जो है इट बिकम्स सॉफ्ट. ओके, सॉफ्ट फ्रूट है दैट मीन्स इट इज ओवर राइपन फ्रूट और उसका शेल्फ लाइफ अब कम हो जाएगा, थोड़ा सा भी धक्का लग जाएगा फ्रूट विल बर्स्ट. ओके, सो ये बीटा गैलेक्टोयूरोनेज एंजाइम जो है उसका एक्टिविटी अगर स्लो डाउन करेंगे देन फ्रूट विल रिमेन nice, turgid for longer time. Okay, and it will not break easily. The over ripening can be slowed down. Okay, so pectin leading to softening of fruit during ripening. Thus, the fruits are easily bruised and damaged on shipment by inhibiting. Underline this, highlight this. इस पे क्वेश्चन बहुत बार आया है. The polycarotouronase by antisense gene. The tomato, which is genetically modified tomatoes, now this is called as flavor saver tomato, but the spelling is different. It is flower sour, but flower sour means it is flavor saver tomato. Okay, so over ripening of fruit is prevented and therefore its flavor is saved. Okay, so flower sour tomatoes can remain on vine until mature and can be transported in firm solid state. Clear? Any doubt? Okay, last application, second last application of plant biotechnology to produce novel biochemicals and vaccines, that is biopharmaceuticals. Plants are potential factories or bioreactors for high value biochemicals such as, highlight this, starch, sugar, lipids, proteins, and products like fine chemicals, perfumes, adhesive compounds, as well as industrial lubricants, biodegradable plastic, and even renewable energy crops to replace fossil fuels. Okay. Now, what is adhesive? From some algae, chemical is extracted which can use to stick cloths together. ओके, सो आगे चल के क्लॉथ का स्टिचिंग नहीं करना पड़ेगा. हाँ, उसका स्टिकिंग करके क्लोथ कैन बी प्रिपेयर. ओके, सो दैट केमिकल इज प्रेजेंट इन सम algae. ओके, सो अगर उसका क्वालिटी, फाइननेस, प्रोडक्शन ज़्यादा बढ़ेगा, okay, so we will be able to stick clothes the way we want instead of stitching them. Okay, so such adhesives are present from some can be extracted from some plants. Okay, biopharmaceuticals are proteins, hormones, antibodies, vaccines, or enzymes isolated from transgenic plants. Some of the proteins that are being produced by transgenic crop plants are human growth hormone with the gene inserted into chloroplast DNA of tobacco plant. Okay, so we will have to consume tobacco to get hormone. Okay, humanized antibody against such infectious agents such as HIV, respiratory syncytial virus, herpes simple virus, the causes of cold sores. Okay, herpes simplex virus जो है that causes cold sores. I am suffering with this now. Okay, and I am taking medications for this herpes simplex virus. Okay, protein antigens to be used in vaccines, for example, patient specific anti-lymphoma, a type of cancer vaccine. Beta cell lymphomas are clones of malignant beta cells expressing on their surface a unique antibody molecule. Currently, many novel products have been commercially exploited for their products, such as a super glue produced by tobacco plants with gene encoding for powerful adhesive protein enable marine mussels to stick to the rocks. It will specially valuable as a biochemical glue for body repairs during surgery. Okay. Transgenic plants containing oil encoding gene from marine algae produce oil that has nutritional value similar to cod liver oil. Okay, so pure vegetarians जो है उनके लिए we have substitute for cod liver oil. Plant that will produce an anti-malarial drug or artemisinin. Genetically engineered opium poppy to produce more powerful painkiller. Poppy seeds. Okay, so these poppy seeds produce some drugs which are painkillers. We have studied same thing in drug abuse. That when they are taken in excess, it is called as drug abuse. Okay, so powerful painkillers can be produced from poppy plant. हाँ, मराठीत ज्याला खसखस म्हणतात. Okay, last part in plant biotechnology. Transgenic plants producing edible vaccines. Genetically altered plants can provide protections to infectious diseases. Plant products act as vaccines, would be inexpensive to produce and thus can easily be made available in developing countries. Okay, inexpensive मतलब उसका research cost है, but once they are produced, once the gene is with plant, okay, it will keep on inheriting in next successive generation. So एक बार research होने के बाद उसका after cost is nil. Potatoes, tomatoes, bananas, soybean, alfalfa, cereals are the most common food proposed for edible vaccine delivery. So raw potato नहीं खा सकते हैं, but raw tomato can be consumed, raw bananas can be consumed. Okay, so these are some of the fruits which can be consumed raw, and therefore they can be used to produce vaccines, edible vaccines. Okay, transgenic animals, many. अच्छा, नेक्स्ट पार्ट का ये बघा. जैसे हमने कहा कि हाउ प्लांट्स कैन बी मेड ट्रांसजेनिक एंड व्हाट आर एडवांटेजेस ऑफ इट. आगे का पूरा पार्ट है दो सर्फेसेस, that why we are producing animals transgenic, why we want to introduce new gene in natural genetic makeup of animal. Okay, so उसके क्या क्या applications है, that is given in next part. Okay, देखते हैं हमारा कितना होता है आज. Okay, transgenic animals. Many transgenic animals such as mice, rats, rabbits, pig, sheep, cows, fowls, fish have been produced through recombinant DNA technology. Out of this, पहले जो सारे दिए हैं, all they are mammals because the results on mammals can be easily extrapolated to humans. Okay. The term transgenic animal refers to animal in which there has been a deliberate modification of a genome, the material responsible for inherited characteristic, in contrast to spontaneous mutation. See, spontaneous mutation will not ensure कि हमें जो चाहिए वही जीन का उसी टाइप का अल्ट्रेशन होगा. ओके, बट बाय इंसर्टिंग द जीन ऑफ अवर इंटरेस्ट, हाँ, वी आर श्योर कि हमारा जीन वहाँ पे है उस ऑर्गेनिज्म में, वॉट एवर द रिजल्ट इज गारंटीड. ओके, so foreign DNA is introduced into animal using recombinant DNA technology and then must be transmitted through a germ line so that every cell, including germ cells of the animal, contain the same modified genetic material. Okay, so all the successive cells or organisms will have gene of our interest. The representative but non-inclusive list of purpose for which transgenic animals have been used indicate wide-ranging applications of the biotechnology. In medical research, transgenic animals are used to identify the functions of specific factors in complex homeostatic systems through over or under expression of a modified gene. See, एक gene जो है, if we insert them in E. coli, okay, so E. coli में उस gene का transcription होगा, translation होगा, protein मिलेगा. Okay, but when the same gene or some new gene is introduced in, let's say, mice. Okay, so mice may, along with this gene, there are many complex genes. Okay, E. coli का genetic material जो है वो it is very small. Overall metabolism is very simple. As compared to that, mice का metabolism is complex. Mice का entire genome is complex. It is large as compared to E. coli. So ये जो हमारा gene है, इस gene का in presence of all other genes or various metabolic activities, how the gene will get expressed. Okay, so उसका complex ये जो complexity है, that can be studied only when it can be inserted in these animals. Are you getting it? Are you getting it?
Yes.
Okay, and therefore we need to modify some animals so that the expression of gene can be studied in complex environment in complex animals. Okay. Next, in toxicology, as responsive test animals, that is detection of toxicants. Okay, so toxicology means some chemicals are toxic. So ये जो toxic chemicals है, इसका effect कितना, at what level it is toxic, what are its effect, that can be studied in animals by making these animals what can we say, susceptible or sensitive to that chemical. Okay, so normally एक जो chemical or the toxin which is under study, mice are maybe resistant to that particular chemical, but we can artificially make these mice sensitive to this toxin so that when the mice is sensitive, ये toxin का mice के body पे क्या क्या effect होता है, that be studied quickly. Okay. In mammalian developmental genetics, in molecular biology, the analysis of regulation of gene expression makes use of evaluation of specific genetic change at the level of whole animal. So एक जीन अगर चेंज किया तो उसका इफेक्ट ओवरऑल जीनोम में क्या होता है. ओके ये जो जीन है इज इट सप्रेस्ड बाय सम अदर जीन. ओके और ओवरऑल मेटाबॉलिज्म अल्टर होता है या कोई एक बायोकेमिकल पाथवे अल्टर होता है. So because of change in one gene, how the effect is on overall metabolism of animal can be studied. Okay. In pharmaceutical industry, targeted production of pharmaceutical proteins, drug production, and product efficiency testing can be done on these genetically modified animals. In biotechnology, as producers of specific proteins, genetically engineered hormones to increase milk yield, meat production, genetic engineering of livestock and aquaculture affecting modification of animal physiology and or anatomy, cloning procedures to reproduce specific bloodlines, etc. Okay. So normally milk yield जितना है by genetic modification we can increase milk yield. We can increase the tissue present in or meat productivity of some animals, or maybe egg producing capacity of some egg layers can be increased. Developing animals specially created for use of xenografting. What are xenografts? xenografts मतलब organ transplant ही है. देखो a tissue graft or organ transplant from donor of different species to the recipient. वहाँ लिख के रखो xenografting. xenografting मतलब एक ऑर्गेनिज्म रैबिट का आय लेके व्हाट दैट बकरी में लगा दो. दैट इज कॉल्ड एज xenografting. ओके.
Basically, organ transplant. ह्यूमन्समध्ये पण केलं तर तेच होईल ना, हार्ट ट्रान्सप्लांट.
होऊ शकतं का त्याचा स्टडी करायचा आहे. पण डायरेक्टली ह्युमन वरती नाही ना आपण करू शकत.
होतं ना.
हार्ट कोणाचं दुसऱ्या स्पीशीजचं. हा
ओके.
मग काय सांगितलं रॅबिटचा आय काढून बकरीमध्ये लावायचा.
असं नाही मला वाटतं.
ह्युमन टू ह्युमन नाही त्याला झेनोग्राफ्टिंग नाही म्हणणार. झेनोग्राफ्टिंग मीन्स ऑर्गन ट्रान्सप्लांट विदिन ऑर्गॅनिझम्स ऑफ डिफरंट स्पीशीज.
आपण जर काही प्लांटवरती आज अजून ह्युमन्स आहेत तर ना वेगळे स्पीशीज म्हणू शकतो का?
कुणाला? हो. आता ह्युमन सोडून दुसरी ह्युमनची वेगळी कोणती आपण आहोत तीच एक स्पीशीज आहे ना. अजून दुसरी कोणती स्पीशीज नाही आहे ना ह्युमनची. नाही आहे प्रॅक्टिकली नाही आहे. देयर इज ओनली वन ह्युमन स्पीशीज. सो ह्युमन सोडून तू चिंपॅन्झीचा हार्ट आणून जर लावला असता दॅट इज झेनोग्राफ्ट. ओके. ह्युमनची नाही आहे दुसरी कोणती स्पीशीज. झालं सगळं जग उलथापालथ करून शोधून आपलं झालं. ह्युमन्सची एकच स्पीशीज आहे आर्या. ओके. एक ही पार्ट करते हैं ट्रांसजेनिक माइस एंड कैंसर रिसर्च एंड देन वी विल स्टॉप. Through laboratory investigations with transgenic mice that have been modified using particular oncogenes. So naturally mice does not have cancer, but we want to study certain drugs on cancerous mice. So artificially insert oncogene, that is the gene which is responsible for cancer. उस जीन को artificially डाल दो mice में, make that mice susceptible to cancer, and now you can test some drugs or whatever you want to on those mice. Okay. So oncogenes and thus develop a certain type of cancer in mice. Questions concerning the relationship between oncogenes and cancer development could be answered. Theoretically, such animals are also used to research into cancer treatments and preventing malignancies. Okay, so particular disease के लिए हमने medicine बनाया. So we cannot test directly on humans. Okay, so वो disease पहले mice में या फिर ऐसे organisms में introduce करो. Okay, so generate करो वो disease and after that the drug can be tested on this disease. Obviously, it has legal aspect. Okay. In laboratory of Philip Lider in Harvard, the transgenic mouse model for investigation of breast cancer has been developed. The oncogene MYC and RAS are analyzed to find out if they lead to breast cancer in mice transformed with these genes. Okay, so हमें जो लग रहा है कि these are oncogenes, these genes are responsible for cancers. So really they cause cancer or not? वो जीन को निकालो, mice में डाल दो, observe mice whether they develop cancer or not. Clear? Okay. Next part, transgenic farm animals. We will discuss on Wednesday, Friday. Okay. So Friday ko this chapter will get over probably in half an hour, and we will start with last chapter, that is conservation. It's all about. ठीक है?
मला एक विचारायचं
हाँ बोल.
होतं.
हो येतोय. बोल.